Today is Friday, the 12th of December, pre-holidays. I really appreciate all who can join us today. And today we have two speakers from Cargill, Brian Lefebvre and Jason O'Brien. And I'll do a short introduction and then we'll hand it off to them. Brian has been doing biotechnology research for quite a while, and from what I understand, he loves doing it. He is director of R&D fermentation at Cargill, and he is part of a team that works to improve commercial fermentations and launch new fermentation processes. In his previous roles, he was in industrial biotechnology, also served as faculty in chemical engineering, also a member and co-founder of a startup in biophysical analysis equipment area. He has bachelor's degree in chemical engineering from the University of Minnesota, Twin Cities, PhD in chemical engineering from the University of Delaware, and his postdoc research was in biochemistry and biophysics at the University of Pennsylvania. Jason O'Brien, our next speaker, is a biotechnology leader specializing in fermentation and biochemical manufacturing. As Senior Director of Fermentation R&D at Cargill, he drives R&D and process development, enhancing efficiency and reducing costs. He's held previous roles in operations, process engineering, in a number of biotechnology processes, from large-scale established businesses to new to the world scale-up. Jason has degrees in chemical engineering and biochemistry from the University of Minnesota, Duluth. And with this, I'm handing it off to Brian. Brian, welcome, and I believe you are the first to start.
Thanks so much. Thanks for the welcome. Thanks for the invite. Thanks for the introduction. Thanks everybody on the call for attending. I did a check beforehand. I believe folks can see the slides. The first slide is just our title slide explaining that Jason and I are going to spend time today speaking with you about fermentation at Cargill. There's kind of two pieces of that. It's only a three-word title, but there's two pieces, the fermentation piece and the Cargill piece. So we'll start off with some information about Jason and myself that kind of supplements the introduction that Irina gave. And then we'll talk about Cargill, big picture Cargill, all the way down to what we do in fermentation at Cargill. So I'll advance the slide. That's me. You can see me on the video. You can see the picture of me up there. When Irina gives the introduction and you hear about yourself, sometimes you think, wow, it was a long time ago I did those things. So I started off in biotechnology. I got my bachelor's degree in chemical engineering from University of Minnesota, and I started doing research in the biotechnology area after my sophomore year as an undergrad. I was fortunate enough to find my way into a laboratory where they did organism construction and fermentation R&D, and I just really liked it. So that kind of set my mind on the path to go to graduate school, where I went out to the Mid-Atlantic region to get a PhD, still working in chemical engineering, but on the biotechnology side of that. And afterwards went and did a little bit more in the biology side, structural biology by proteins by NMR. And part of the work I did there led to the launch of this startup company, which is still functioning, selling equipment, specialized equipment for doing protein NMR under really high pressure. So I work with some really smart people who are really good at the structural biology piece, and I kind of brought the engineering side. How are we going to design devices that allow people to do this kind of work? Before going into industry, I was a faculty member at a school in New Jersey called Rowan University in the Philadelphia metro area as well. So I spent 5 years as a faculty member there in chemical engineering, teaching and doing research in biotech and pharmaceutical area. And then was lured away from academia to go work at experimental station in Wilmington, Delaware for DuPont, where I spent about 10 years there. First 2/3 of it was doing cellulosic ethanol fermentation research, which is a process that during my time there, we took from laboratory to pilot to demonstration scale with a plant in Tennessee that I did a lot of experimental work to inform the design and help start up that facility. And then kind of the same process over again to get plant built just outside of Ames, Iowa, in Nevada, Iowa, a commercial scale plant. So it helped a lot with the design, experimentation, startup of that facility as well. And then eventually at DuPont, also got into other types of fermentations and team leadership and competency development. And then not quite 10 years ago, came back to the Midwest to work at Cargill, still working in the fermentation space. working here out of what we call the Minneapolis R&D Center, which is in Plymouth, Minnesota, which is a western suburb of the Twin Cities. So we have a fantastic team here, a fantastic facility here for doing fermentation research, and we'll tell you more about that after Jason introduces himself.
Thanks, Brian. So similar to Brian, you can see me. You can see my picture up on the slide deck. A little different. You see me in an office now. That was interesting day in the plant. As you can see some stuff on the wall behind me. Tying back to Iowa, so my first experience in biotechnology was with Cargill in Eddyville, Iowa in like 2004. So I'm just over 20 years now. I spent a lot of time in Iowa working for Cargill. I worked at our Eddyville plant a couple of times. I also worked for another company and was based out of Ames for a while. Both of my kids were born in Iowa, so I'm actually really happy to be talking to a group of folks that have a good presence in Iowa. As the introduction said, I've done a number of things through my career, really focusing on different levels of operations management and like process design, culminating in my current role where I'm leading up our fermentation capability in the Cargill R&D group. So very much looking forward to the conversation we're gonna have today.
Great. I like how Jason pointed out, Jason's picture shows what it might look like to work at a fermentation plant facility. You can see him with a hard hat and a tote behind him. And I actually thought it was like an artistic design on the wall and not a bunch of stuff that had splattered out of a pipe somewhere. Whereas with my picture, that's a picture of me when I was doing a tech transfer to a commercial manufacturing site. That's what R&D people do when they go to manufacturing sites. They go and eat in fancy restaurants and occasionally show up at the plant and see how the process is going. Anyways, let's talk about Cargill to start. For those who don't know, Cargill is a gigantic company, private company, maybe doesn't do a lot of publicity, but is one of or the largest private company with 160,000 employees worldwide, operating in 70 countries with, I think we can now say 160 years of experience. I don't know when the first facility actually opened in 1865, but we're so close to the end of 2025, we can say 160 years of experience. And revenue, a recent year, a revenue of $160 billion. There's a gigantic company whose overall mission, if you were going to say it in one word, is to nourish the world in a safe, responsible, and sustainable way. That's a really, really, really big area. So we'll spend a little bit of time talking about just how big that is and eventually start to focus in on how fermentation plays a role in that. But you can think about how Cargill connects farmers to markets, provides ingredients for customers, gets essentials into the hands of consumers. And this slide is a little bit busy, but it shows some of the ways that Cargill operates across the agriculture enterprise, from partnering with farmers to grow crops, raise animals, bring those into the value chain, turn those into food products, transport those food products around the world. So you can start all the way on the left. And in the middle, you see some examples of areas we play in: meat, egg, salts, oils, starches, cocoa, sweeteners. The bottom of the center gets a little bit more to the fermentation specific stuff when we talk about creating nature-derived bio-based products and biofuels. And on the right side, you can see how we move that from farm or refinery, biorefinery, into our partners, companies we partner with, who then turn that into food products and ingredients which go out into the marketplace. So Cargill has tons and tons of capabilities in a lot of different areas. We're coming to you from an R&D perspective, but working in R&D is fantastic, and it's also fantastic to work for a company that has all of this commercial capability. So when we have successful R&D, we can actually get into a production environment and lead to the creation of new products that get into customers' hands. When we were kind of talking with Nigel about what we want to cover in this, and we learned it was an Iowa-centric audience probably, so we created a map. Jason created a map that shows the locations that Cargill has, the major locations that Cargill has in Iowa, with a little graphic that shows, grain terminal versus a corn processing plant versus a soy plant versus an egg facility versus a postbiotic facility. And the picture is a rectangle. Iowa is not quite a rectangle, so there's parts of neighboring states that show up on this rectangle as well, but you can see that Cargill has quite a presence in the Iowa area. And we'll talk more about the corn processing plants and how that links up with some of our fermentation sites in Iowa in the coming slides. So we're starting our move from very, very broad into the agricultural commodities that lead to fermentation inputs. So why do we have plants in Iowa? Well, Cargill was founded in Iowa as a grain business with a single grain elevator, and now has grown through successful business practices and expansion to be a major player in domestic, international, renewable raw materials markets, where, like I said a couple slides ago, we bring in raw materials from farmers and ranchers, and we turn those into food, feed, energy, bio-based materials. There's waste streams that are created as part of that, and we work to use those within the plant or to put them back to farm, forestry, agriculture uses. And as we get a little bit more into how those agricultural feedstocks turn into fermentation feedstocks, the nugget on the bottom left, the Midwest, Iowa, for sure, other areas of Midwest, huge in corn production. 86% of the corn harvest in 2023 was from the Midwest, and that's 13 billion bushels of corn. A bushel of corn is an 8-gallon bucket, or about 55 pounds. So that is a lot of corn that Cargill buys, processes, turns into products, some of those products being things we use in fermentation. So Jason's going to talk a little bit more about corn.
Yeah, thanks, Brian. We can jump to the next slide. I guess one thing I will say to kind of bring back what Brian was saying there, that last slide, Iowa really is a key focus area for us within the bioeconomy. So we talked about corn, and we're absolutely going to talk a lot about corn in this conversation. But if you go back to the map, we also had soy processing, biodiesel plants. We have a Cargill protein plant that focuses on eggs up in Mason City. So we have a fair amount of other types of products that we also make in the state of Iowa, capitalizing on resources and talent. But yeah, for what Brian and I do, right, coming out of a fermentation background, we're going to talk a lot about corn, and we're going to talk a lot more about a fermentation type of plant. So starting with this, staying with the kind of bioeconomy theme, looking at raw materials. So when you think about things that you can feed into fermentation, you can look at corn, you can look at wheat, you can look at sugarcane. All of them have the ability to take a carbohydrate and get it to a simple sugar. We obviously grow a ton of corn in Iowa, so being that we're talking to more of an Iowa-centric audience, we tailored it that way. We do use both other feedstocks globally within our fermentation product lines. So why is corn a good fermentation feedstock? It really comes down to the starch. So roughly 63% of corn as you receive it, whether you're at an ethanol plant, you're at a co-op, you're at one of our facilities, roughly 63% of that is going to be starch. And starch is a polymer form of glucose, right? So you can break it down into its monomer form, and you can have it as a simple sugar to feed into fermentation. I have another slide coming up that's going to talk about the other things that we do with corn at our plants. If we want to jump to that, Brian.
Sure.
So this graphic is not from me. It's from the Corn Refiners Association, of which Cargill is a member, as well as some other large corn processing companies. This is a diagram of essentially how a wet mill works. So you bring in the dry grain, you steep it. That is effectively soaking the corn. It softens up in the period that you're holding it in its steep. And then you go through a process of mechanical separation to take the components of the corn and really try to maximize what you do with them. So one of the first things you break off of the corn, if you're looking in the kind of upper right corner, is the germ. So you will separate the germ away from the starch and the fiber. That germ is where the majority of the corn oil is stored within the corn. So at our wet mills, we actually separate out that corn oil to be used in food applications, and we take the remaining corn germ and we use it as an animal feed. Your next step down the line as you work through the left is you pull out the fiber. So the fiber, right, generally ends up in animal feed. There's not a whole lot of other uses for it, at least in traditional senses. If you look at some generation 2 type feedstocks, you can start talking about cellulosic availability within some fiber streams. Not something that we do today. we send it all into animal feed. As you continue down, again on the left, your next big component is the starch itself. So you get starch and gluten off at pretty much the same time. You separate those out. The gluten goes into a feed. You will hear something called corn gluten meal. It's a high protein feed product that gets mixed in with dairy cattle and other animals. Then the other big piece you're getting out of that is the starch itself. So we do multiple things with the starch across our plants. Some of it gets converted into a high fructose syrup or glucose syrup, depending how you want to call it, that gets added as a sweetener into foods. For our discussion today, we're going to talk a lot about the starch that gets turned into dextrose or glucose, though, and then gets used into our fermentation plants. Next slide, Brian, please. We put a very, very simple process flow diagram in this presentation just to talk about the fermentation plants a little bit. So really common setup, some sort of media preparation, right? You're going to add a media into your dextrose to grow your organism. It's kind of like growing plants in a way, right? You're gonna have some nitrogen source, you're gonna have some phosphate source, you'll add a couple other traces to make your organism happy. You'll go through some amount of seed train to build up your culture quantity so that you can make the best use of the fermenters that you're in. You'll end up with a product that has pretty high water content for the type of fermentations that we do. So you'll go in and do some separation where you'll get your biomass away from it. We have a couple types of unit ops that you might see in that separation. You'll go through purification, which can look very unique depending on the process that you're in, right? You can have things like ion exchange to remove residual ions from the nutrients that you added. You can have separation based on charge or molecular weight of your product. You can do separation by phase. You could have an extraction part of your plant, again, depending on what you're making. The next step is really the finishing formulation part of a process. So if you're making a liquid product, it might be what concentration you're bringing it to. If you're making a solid product, you might be going through a crystallization step or a classification step to make sure you're getting the right sizes. You may add in some additives for storage or for shelf life. And then your final step in all of these is really packaging and storage. So if you're making a consumer type good, you will have potentially a really small package. If you're making something in bulk that goes to like a food manufacturer, you might have something in a rail car scale, or you might have something in a truck scale. Very dependent on what you're making and what makes sense for that process. Next slide, Brian. Okay, perfect. So I have two slides here that kind of feed off each other to highlight one of the ways that Cargill does business in the space that is fairly unique and capitalizes on us both being a corn processor and a biotechnology player. So we have biorefineries around the Midwest, 3 of them that we highlight. 2 of them happen to be in Iowa. So I mentioned at the beginning of my presentation that I started in Eddyville, Iowa back in 2004. That was our first, what we call a biorefinery. So integrating a corn processing plant with biotechnology plants. We then built one in Blair, Nebraska, right over the border, just north of Council Bluffs. And then our most recent one in central Iowa, north central, is in Fort Dodge. So about an hour and a half north of Ames. If you jump to the next slide, Brian. So this is a top-down view of our plant in Blair, Nebraska. And I'm going to talk through, I should have had a pointer. Is there a way to turn a pointer on this? I don't know if there is. So apologies, this might get a little hard to talk through. But our biorefineries partner both internal and external to Cargill. So if I start at the picture on the right, if you go to the lower right corner and you see some silos sitting next to the road, yeah, sorry, I should have thought that through better. We have where the corn comes in. Let me see if there's a way I can get a pointer on here. I don't think I can. Okay. So if you were to go to the far right of the picture, you can see a couple really big silos. That's where corn comes into the plant. If you go, yep, right about there, Brian. If you go north of those, that is the mill. So Those silos can handle the plant for roughly a week. So we have multiple millions of bushels of on-site storage. We design that way so that if we have bad weather come through, because that sometimes happens in the Midwest, you can keep the plant running when you're working on cleaning up roads and whatnot. So the corn comes in, goes north, which is the mill. That's where we do, if you think back to that diagram I showed, that first kind of break off of the feed versus starch components. To the right of that mill is where we have our extraction plant to pull out corn oil. If you go back to the mill and go north again, so up, that is the refinery. So that's where we are converting that starch into dextrose. And from that point, it goes out into the broader plant. So if you go directly left of the refinery, yep, right there, that is our ethanol plant in Blair. That is a large 200 million plus gallon ethanol plant. It is one of the larger ones in the US. And then I'll just speak to the rest. If you look at everything else in this picture, those are co-location, either partners or direct Cargill facilities. So in the upper left-hand corner where Brian was a minute ago, that is a plant that I ran in Blair for about 5 years, where we do an internal fermentation. We make erythritol there, as well as a newer product, EverSweet, which is a high-intensity sweetener. As you move to the right, we have an internal lactic acid plant, which is the largest in the world. And that closes up the Cargill exclusive ones. You can go online and Google who's in Blair, Nebraska, and you'd see our other over-the-fence partners. But in general, with public conversations, I try not to call them out as where they are. But the model is the same really for all of them. We get to a dextrose or a sugar feedstock, and we provide that to the customer or ourselves. And if you look in the lower left-hand corner, you can see a diagram of how that works. So what we try to be is we try to be a good partner where we can help with some of the utilities, some of the water treatment, potentially some of the talent, to reduce the investment cost for someone trying to have a unique biotechnology process. And then we also, by having someone partner with us, we provide a location where we can provide dextrose without having to have some of the shipping costs. So it turns into a nice synergy where we can create these concentrations of knowledge and reduce costs for all the parties that are participating. Okay, Brian, back to you.
Okay, thanks, Jason. This is a slide that Jason made with this timeline, but I get to talk about it. Cargill has a really large fermentation footprint. Like Jason said, our first build was in Eddyville, Iowa. Our most recent build is our Eddyville, Iowa site. If you look at this timeline, there's a color code to investing in corn processing plants and then investments in fermentation assets at those plants, co-location partners joining us there, or joint ventures joining us on those sites. So over $2 billion invested globally in fermentation processes at Cargill, starting with 1967 was the first corn mill that Cargill started in Cedar Rapids, Iowa. And for 20 plus years, Cargill milled corn without any fermentation. In 1990, right here, you can see this one with the star over the top. It's the first commercial fermentation for Cargill. It's a citric acid fermentation. Was the first one to launch, and then you can see throughout time other product launches. There are future slides that talk about the different, the variety of fermentation products that Cargill produces. Well, that's sort of the beginning side of milling corn and then getting into doing fermentation. And then over here towards the back end, the most recent one, and maybe you've seen press about it recently, at the Eddyville, Iowa site, we recently commercialized with a partner. It's a JV called Core, where we ferment the kinds of things that Jason just talked about, the sugar that comes from corn, plus other nutrients and goodies for the cultures. We produce 1,4-butane-diol with that joint venture called Core, which is an alcohol, but is a feedstock for making polymers, which can go into fabrics and apparel. So lots and lots of investments over the last 35 years in the fermentation space as we launch new fermentation processes and improve existing ones. So how do we do this? Why do you have two people from R&D talking about this? We've talked a lot about big Cargill. We've talked a lot about what happens at plant sites. Where do we in R&D fit into this? Or what I call turning a concept into reality. How do we take something that is an idea in the laboratory and make it a commercial reality? Cargill is a business. Cargill likes to make money. So we kind of first have a filter for, is there a product we can make in fermentation that will be more valuable than the feedstock that we're putting into the fermentation and the money it costs to build the assets, run the assets, and that sort of stuff. So there's that money side of it. But if you talk about the technology side of it, there's this concept of a manufacturing readiness level. That's the general concept. Recently, Biomade, some folks from Biomade developed what they call the biomanufacturing readiness level. You can read about it either through Biomade or find the paper in the literature. It's this concept of, if you have an idea, how do you advance it through stages to get to the point where you can commercialize it? And that's what we do in R&D. With the different fermentation products we have, we've been involved in some of them. It's been all the way from manufacturing readiness level 1, where we are, Cargill overall, or our partners on other science teams at Cargill, are proposing a concept that has some scientific basis, and we partner up with them to prove that out in the laboratory with experimental proof of concept, validate it in the lab environment, and then work towards designing a production site, optimizing the process to fit in that production site, launching that commercial site and getting capability and rate in place, and then getting to full production process for the whole plant site. And other technologies, like the most recent one with Core to produce 1,4-BDO, we come in, we partner with somebody else to make a JV, and they've already maybe at level 3. And we still work with them to bring it in-house, prove the technology out in our hands, and then they get to work with the other folks at Cargill on figuring out how to build, start up, staff, successfully operate the plant for profitability. So I would say that our laboratory and pilot infrastructure at Cargill allows us to move new concepts from idea to commercial. And I show here just sort of what I consider in fermentation the beginning and the end points. The beginning point being, what I feel is the smallest equipment we use, which is a device called the biolector, which allows you to conduct on the order of 1 milliliter size fermentations with pH control and feeding and sampling, all the way through running that same fermentation, but in a much, much larger commercial scale where you're talking about thousands, tens of thousands, hundreds of thousands of liters of production scale. So at Cargill, the facilities we have to accomplish this go from concept to commercialization. I put some pictures, Jason put some pictures here showing how in a fermentation process, there's the strain piece, building organism or partnering with another company to build an organism or bring in an organism. We have the fermentation team at Cargill does not build organisms. We have other groups in Cargill that focus on that. And we work very closely with them to do the genetic modifications that allow us to make new products with organisms. And we call fermentation development. We use a range of equipment from the biolector I showed on the last slide to AMP 250, which is an automated robotic system where the reactors are 250 milliliters, and each individual reactor can have a different temperature, pH, stirring rate for oxygen transfer, feeds, off-gas monitoring, robotic sampling, up to the benchtop reactor scale, where you have reactors that are a couple of liters in size, all the way to piloting capacity. Sometimes things run different at pilot capacity because of back pressure, because of CO2 solubility dissolving, because of the changes in mixing you get as you advance from a small vessel to a large vessel. We have great pilot facilities at Cargill as well, including one at the Eddyville, Iowa plant site, with the whole goal of this process to make products through fermentation that can be put into food products or out into the marketplace, get into the consumer's hands, to make money for Cargill, but also to launch nitrogenous ingredients or useful molecules. Cargill works with the range of products we make. We're fortunate to work with a lot of different organisms. We don't make all of these things with one organism. We work with a lot of different organisms, some bacterial, some yeast, some fungal. We run fermentations that are aerobic. We also run anaerobic fermentations. Some of our products are food, so we have food-grade fermentations. We have other fermentations that are technical grade. We have other parts of the organization that can take the outputs from fermentation, whether it's prototypes made in piloting or market development type batches made at commercial scale, and put them into applications, making drinks or food products with the fermentation ingredients. And there's also the legal and regulatory portion of launching new products, new fermentation processes, putting new equipment on sites. We have talented folks at Cargill that are experienced in building regulatory dossiers, thinking about permitting, all those types of things that get into sort of the nuts and bolts of being able to run a commercial fermentation process. This is a map of the parts of the world where Cargill has fermentation assets. The right side is a map with different colored dots on them. And the left side is sort of the key to the map where Cargill makes alcohols, organic acids, polymers, enzymes, glycosides, and bioactive components at fermentation sites worldwide. In alcohols, there's a lot of ethanol plants. Jason mentioned, ethanol plant at Blair, when we had that overhead shot of the Blair, Nebraska plant. We also have in North America, Fort Dodge and Eddyville are ethanol production plants. We have ethanol production plants in the EU and also in South America. And I mentioned the one for BDO, butanediol, the joint venture with Core. That's located in Eddyville. And then erythritol, sugar alcohol, is made in Blair. Organic acids, maybe 15 years ago, Cargill got a lot of positive press and awards for advancing lactic acid fermentation technology with a new Cargill-specific, Cargill-designed, Cargill-found, Cargill-built organism that improved the efficiency of lactic acid fermentation process. We operate that process out of the Blair, Nebraska facility. Citric acid production in Eddyville, Iowa, and also in South America. We call it KGA, ketogluconic acid, produced in the EU. Polymers in a couple of our European plant sites, enzymes at a European site, and also toll manufactured. Another, not as recent as the BDO, but the product before that was launched in a glycoside called Eversweet, which is 2 Reb molecules that are found in stevia, but instead of taking them out of the leaf, making them through fermentation process, that's commercialized at the Blair, Nebraska plant. And then some bioactive molecules, Epicor and Imin B, things that go into kind of the pre-post-biotic market. One made in Ankeny, Iowa, and one in Cedar Rapids, Iowa. So, like I mentioned in the prior slide, Cargill makes a lot of stuff, and we make it with a lot of different organisms, which makes R&D at Cargill a ton of fun because we have really great equipment and really great people and get to work on a really wide range of fermentation processes, whether it's improving existing ones to make them more profitable, or it's launching new ones to the market. So we've used this image a couple of times to kind of talk about, in the agricultural commodity point of view, how we bring that in at Cargill, how that gets turned into fermentation feedstocks, how we use those to make products through fermentation, but also the other parts of the corn that might go into animal feed or corn oil, how we have integrated plant sites where we can make use of the byproducts and waste streams, how that can recycle back into the farming, forestry, agriculture. Jason mentioned when we had that corn processing diagram up about corn gluten meal and some of the listed here, some other things that come out of a corn plant that can be animal feed. So we're using the parts we need for fermentation. Sugar as a carbon source, and the rest of the stuff, the high protein content, the fiber content, other nutritional parts, either go out as corn oil to the human food market or can go to animal feed type purposes. That's the end of the prepared material. It took about 40 minutes. We're open to questions, whether it's questions on Cargill overall, corn, fermentation, plant sites, questions about Jason's background, questions for me, whatever folks would like to hear about. Ample time for questions. Thanks for your attention so far.
I have a question. Can I?
Yeah.
Okay. Hi, everyone. My name is Esther. I'm a PhD student here at Iowa State University. I work largely with bioreactors and fermentations, and I have about 3 questions. The first one is when you talked about like the fermentation process in terms of like feed strategy.
Mhm.
How do you decide if it's either batch, fed batch, or continuous? And how do you know which is like more preferred for like whatever product you're trying to make? And my second question is, in terms of corn being like a core feedstock, how do you maintain the consistency of corn, it being a seasonal crop, for example?
Mhm.
Yeah. And the last one is in terms of the strain engineering, what would you say drives the decision in terms of whether to improve an existing strain or making an entirely new one?
Great. Thanks, Esther. Good to see you again. Thanks for all of these questions. Jason, I can answer all of them, or you can answer some of them. So speak up and maybe grab the ones you want.
Let's start with probably the easiest one. How do we control corn quality? Short answer is we kind of don't. So there is some variability season to season. So if you look over the years, you will have higher and lower starch content as a general statement, depending on things like moisture, when did it fall, what were your temperatures during the middle of the summer and growing season. So we do see year-to-year variability on the starch content in corn, which will affect the amount of like yield that we get off of a bushel. As far as more tactical quality questions, we do have things like moisture specs and test weight, which is how much mass you have per volume, that gets at like, was it a relatively healthy corn or was it a little more stressed? We also look for things. We also look for potential toxins that can be in the corn to make sure we're bringing in safe products. So we absolutely do have controls, but to the amount of like starch, that can fluctuate year to year and region to region, depending on how the growing season looked.
I think there were also questions on, I guess, what's a fermentation process design? How do you do batch, fed batch, continuous? I can answer that. You can give an answer to it, Jason.
Go ahead,
We
Brian.
can tag team on it. I would say, it's largely driven by economics. And so we want to maximize in fermentation. The job is to maximize the big three: the titer, the rate, and the yield. You want to make a lot of the stuff. You want to turn your substrate into a product with a high yield, and you want to do it pretty fast. And different organisms have different preferences for how they would like to have their feedstocks presented to them. The flip side of that is if it's cheaper to build and run a facility if it's just a batch fermentation. If it's a fed batch fermentation, you have to have the feed tank, the pumps to feed the feed in. You have to have probably more process automation, process control. So Cargill runs batch processes, Cargill runs fed batch processes. So we kind of tailor it to what works best for the organism. Some organisms don't care if you dump them into a solution that has 150 grams per liter of sugar. They'll just get right to work. They'll make things with the same yield that they would if they had 100 grams per liter sugar or 50 grams per liter sugar. So then we say, okay, let's just put all the sugar in up front and let it go. Other organisms, if they see too much sugar, too little sugar, the yields get different. You end up making byproducts versus products. So you have to kind of tightly tune it in to keep it in a range where the organism is doing the stuff you want. And that's a case-by-case thing, because every organism and every product we make is a little bit different. I'll pause there and see if Jason wants to add anything onto that.
Nope, I think that was a great answer.
Cool. And the other one, I think, was a question around organism. How do we decide whether to improve an existing or to go and like launch something completely new? That kind of also goes back to a case-by-case or an organism basis. The reason we have so many organisms is because there are positive features about some of them and negative features about all of them as well. We could not take the organism we use to make lactic acid and expect it to do a good job of making erythritol. That's just not going to happen. So you kind of look for things that are adjacent to maybe what that organism likes to make in its wild native state, and then see, okay, it makes something pretty similar. So we could maybe add on to that, add a couple of genes, make sure we get them expressed at the right level, and then make a related product or make more of the product to kind of supercharge the organism. I'll kind of stop there and see if Jason wants to add anything.
Nope, again, I think that was a great answer.
Very good. And Esther, I hope I answered the questions you posed. I do see other hands up, but Esther, if I didn't address your question, you can speak up and ask again.
No, you did. Thank you.
Different thing.
We have a question in chat.
Yeah, so there's a question in chat. I'll go ahead and jump in with my somewhat cheeky or introspective question, but so one of the things I struggle with, right, is how much plastic waste my own lab generates, right? Like all the tips and all the pipettes and all these things. We're using all this plastic in order to reduce reliance on petroleum, right? And so it's kind of the same question about those tiny little fermenters, right? In terms of they're really great and they obviously save cost in terms of instead of having to do the big fermentation, right? You can start smaller, but then none of those are reusable, right? And so where do you determine the trade-off, right? In terms of those little reactors are so expensive, and do you just kind of go by feel, or is there like a model that you use?
So I can talk about this for a while, but Jason, go ahead.
Yeah, so I don't know that there's a model. To your point, Laura, we're also sensitive to that, right? There's a lot of waste. B, they're expensive, but they're pretty powerful in some of the early data you can get. So they are one aspect of tools we have, right? So we showed the bioelector. We've talked about some of our high throughput. We had a picture of that. We have traditional like 2-liter capacity, right? So glass reusable. We have piloting assets. So I think you have to balance what question are you trying to answer. You have to balance the efficiency of getting to that answer, and you have to look at the cost that you're using to get there, and just make sure you're being responsible with the resources that you use. I wish I could give you this is the one size fits all answer, but I don't think it exists. Similar to some of the answers Brian was giving about process design, I think it's a little bit case by case.
I'm going to say, thinking not really cynically, but they cost money. So, and to me, the cost is a strong deterrent, a strong enough deterrent. Yes, we would like to have less plastic waste as well. But it's a 250 milliliter reactor. If we run a full bank at 24, I think most people probably recognize fermentations are not done in one day. So usually most of our fermentation processes are multiple days or even up to, even 2 weeks at an extreme. So you're talking about making 24 pieces of plastic that are about the size of a soda can getting generated every week or every other week. It's not nothing, but it's not a ton either for the amount of information we get out of it. And then like Jason said, we also eventually advance the concepts, the winners from that kind of experimentation into our reusable vessels at the 2-liter, 20-liter pilot scale. And I would certainly love it if we could put reusable reactors into the Ember 250 system. But I have not been able to find that yet.
And the tubing and all the packaging that came in, all that, right? So yeah.
That's one of my favorite lab jobs, is breaking down the cardboard boxes that these things come in. Thanks for the question.
We have the first question that was in there was around some of the uses of our products. So I can certainly highlight some of those. So lactic acid is a good one, since we just talked about plastics. So our lactic acid plant in Blair, Nebraska, we're actually building another one internationally right now, but that goes to polylactic acid. So we feed a partner organization, NatureWorks, with our lactic acid, and we create a biodegradable plastic. Lactic acid can also be used in food preservation and a lot of food applications, but that plant was initially purpose-built for the plastics business. Citric acid is another one that we had highlighted earlier in the presentation. So that goes into a lot of candies. Again, a lot of food preservative because it has low pH. Gives you the sour taste. We talked about erythritol earlier in the presentation, right? So that's a low calorie sweetener, also has some bulking components to it. So you'll find that in a lot of products out there. BDO is our newest one. So that is a drop-in for previous versions of BDO. So you see it applications like spandex and other things. That plant's been online for roughly 6 months. So that's an example of some of the places our products go. We have a number of ethanol plants that go into either fuel ethanol or potable internationally. Everything we have in the US is fuel. If there's other questions on where they go, I can certainly answer, but hopefully that covers kind of the broad brushstrokes of where they go. See some hands up too. Maybe do another hand before we go to the next one in chat. I think Florence, you might have been the first one with a hand up.
Awesome. Sorry, I'm having issues with my video. Thanks so much for your presentation. So the first question is way back at the beginning, when you were talking about the use of the fiber that you separate, it was back into the feed. And you mentioned that there is some potential there. And I'm wondering if you could talk about from a business perspective, is there interest in developing that into using the cellulose in other ways, developing that into cellulose-based materials or things of that nature? Or is it that there's so much demand for that fiber in the feed that you would probably source cellulose from somewhere else and you probably wouldn't use this fiber from this process that way?
That's a really good question. I'm going to do my best not to butcher it and not have someone look at the recording later and coach me on how I answered it. So I think where you got to go to, Florence, you're kind of asking the question, is really the value proposition of what you do with it. So right now we have a well built out feed business. It certainly creates value. We have a lot of good partners in close locations to our plants in the Midwest, as well as down in like the Texas area. So we certainly have good outputs for all of our fiber today. We also have a history of looking at both traditional and maybe next generation carbon sources for fermentation, even though we've really only commercialized the traditional sources. So I do believe that if we found the right value proposition in the future, whether we looked internally, which I think we would do to understand the cost, or we looked externally, I think we would also do that. But yeah, again, today we're primarily the traditional sources.
Yeah.
And to your point, yeah, we have found ways to use as much of the corn as possible. So we have good feed outlets for that fiber right now.
Yeah.
Does that answer your question?
It does. And then the follow-up, if you have time, you can get to other people's questions first, but I would love to know what happened in that interesting day in the lab where you had the splatter in the background.
Yes. So that was in a plant setting, and we had a little bit of a leak that we found a little bit later than we would have liked. That's what it was, and it was a fun picture for me to send to someone of a day in the life of the plant when we discovered something. So, yep, but that's what it was.
Well, thank you so much.
Mm-hmm.
We also have, if you go to the office center for Cargill, which is a little bit south of where we work, it's a modern looking interior building with glass panels, and sometimes they have, abstract designs on them. So maybe that was the inspiration for some of those designs. It was this picture, Jason, of yours there.
We could probably reapply it. Daniel, you have your hand up.
Yes, thank you for the presentation. I guess my question would be, it was in your slides when you're discussing going from the smaller scale to larger scale.
Yeah.
I feel like you're not always going to have 100%. It's going to work every single time. Could you discuss a little bit about the troubleshooting you do?
Sure. I'm trying to get to the right slide here.
That's it.
That's part of the fun of experimentation is we do our best to come up with good experimental concepts, but not all of them work out. And ultimately, we're on the hunt for the best fermentation process we can run. It will generate the best mix of titer, rate, and yield to drive that techno-economic analysis to lead to the most profitable commercial operation of these processes. So we try a lot of different things in the lab, and not all of them are equally successful, but they are all great ways to learn about what was a step in the right direction, what was not a step in the right direction, where should we drive into the future. So at the lab scale, I don't really think of them as failures. Contaminations are probably failures. But if you set up an experiment at a different pH, a different temperature, a different feed rate, a different dissolved oxygen, a different stirring scheme, whatever, not everyone is going to be the best one you ever did, but you're starting to develop a map of where is the best that I know of so far, and how can I use that information to inform where I want to move in the future, where might be the future optimum. It's like an optimization problem. So you can apply design of experiments, statistical type techniques to help you design those experiments, analyze those experiments, push you in a new direction, use your past data to understand what might be successful places to look. So that's part of the fun of being in the lab. By the time we take something to a pilot, we'd like to really have proven that particular process out at a pretty high degree of confidence. So then because we want every pilot run to really be a success. And if a pilot run isn't a success, it's maybe because of contamination or because a piece of equipment that we thought maybe had a certain type of capability didn't quite have the type of capability we thought. But the process itself, like temperature, pH, all those sorts of things, should already sort of be locked in, and you're really just testing to see that the pilot equipment can deliver those things, and if anything unusual pops up because of the scaling difference. And then at commercial scale, we need everything to be a successful run. We can't really it's a big problem to waste an entire fermenter full of liquid with not a lot of product in it. So hopefully I kind of cover the spectrum from lab scale. We have lots of not so successful runs that are helping us understand where to move to be more successful. Then through the spectrum of piloting and commercialization.
That was a good answer. Could I ask one quick follow-up question for the
Sure.
lab scale?
Yeah.
I guess how many runs do you imagine do you find yourself doing before you just give up? If you ever give up.
Give up? No give
If
up.
you ever give, no give up. All right.
So in industry, if there's a commercial case for it, we keep working on it. We don't say, oh no, that's the end of the road. No, we always have new things to explore. There could be new strains coming from our biology partners. There can be other spots in the process parameter space to look for, temperatures, pHs, feed rate, dissolved oxygens, all sorts of things like that. So I would say commercially, the only reason we maybe really stop something is if the commercial case erodes. Somebody says, ooh, 3 years ago, we thought we could sell this molecule for this, but now it looks like the market is maybe not in that space. So we won't make any money making this thing, so you should probably stop doing research on it. That's an example of where we stop, but it's not stopping for a scientific reason. It's more stopping for a business reason. Jason, if you want to give a different perspective, correct me at all, you can.
No, I think that's a fair answer. We work on things that have value propositions attached to them. We love technical challenges, and we will sometimes chase the same hill for quite a while until we have something change in the value proposition. Jumping to the next question before I get bumped off because we're getting short on time. There was one question in the chat real quick. Do we use order of magnitude estimations for cost estimations, early stage analysis? Brian's referenced TEA, techno-economic analysis, and I've talked about value propositions a couple of times in this presentation. Critically important. There has to be a value. There has to be a line of sight into how this creates value for our customers. How does it solve a problem? So yes, we are very big on that. And I see one hand up with a few minutes to go, so maybe we get to that question yet.
I guess it's me.
Yes.
Hi, Brian. Hi, Jason. It's nice to meet you here. Yeah, so my question is, like I just create a case. Like you talk about organic acid. So if we have one organic acid, we already do the fermentation at like 500 liter, right? In that institution, right? In order to attract collaboration potential with some company like a Cargill, what should we do next? Should we continue to scale up, try to improve the titer yield productivity, or we should do something like downstream processing or like TLC? What should we do in order to like collaborate with the Cargill?
I'll let Jason take that first.
It's a good question, and like some other things we said, I think it's going to depend. So value proposition is key. So you have a novel molecule, you have a novel product. Where do you think it will fit in? What sort of problem are you solving? What do you think the value of that organism is, or that process would be, right? It's going to cost us X amount to make it. So a rudimentary TEA, we think that has a market value of this, which starts to give you insight into, is there a value prop there to be had? The next comment I would have is it goes into, once you define your value prop, there is that relative maturity of technology that Brian covered in the PowerPoint. Certainly, the more mature you get for at least general idea of unit operations, some idea of where you think your scaling difficulties would be. So if it's highly aerobic, right, you start talking about vessel geometry, and once you get big enough, you have enough head pressure that like solubilities start to play. Like if you can start identifying your risks and what hurdles you need to get over, and then like how you're planning to go after them, those are also things that become more powerful in a proposition to a large company of like how you can partner and how you can try to bring something to market. Those are like top of mind, but again, it's kind of a wishy-washy answer. It really does depend. Start with a good value proposition and then figure out where you think your big hurdles will be. And then I think that starts you off in a pretty good place to have conversations. And I'll hand off to Brian for anything I missed.
No, it's good. I would just maybe make it an even bigger picture. When I think about people, or when I talk to people in the group about making new bioproducts, I kind of separate it into two cases. One case, you're going to make a molecule that's already on the market through a petrochemical means, but you want to make it through a biological means. That's great. You need to understand what the economics are for the petrochemical case and then come up with a biological process that matches or betters that. Yeah, sometimes it can be a little bit worse economically, and people will pay an upcharge for sustainability or bio-derived, but that kind of gives you anchors for, okay, I better have a process that at least gets me in the ballpark of how they can make this molecule currently. The other case is you're making a brand new molecule. You can charge whatever you want for it, but nobody knows what to do with it. So there you really kind of have to work more on the market development side. What would they use this molecule for? Do I have examples of what we could turn this molecule into, or who might buy this molecule, and how much they might want to buy? So that's kind of more of the game over on that side. And yes, the technology is important, but the technology really doesn't matter if you can't make a case for why would somebody want to have this molecule in the marketplace.
Thank you. That's good answers. Thank you.
Thank you for the question.
Okay, I think that's all the time we had today. Thank you very much for spending your Friday afternoon with us. We have good questions. Today is Friday, the 12th of December, pre-holidays. I really appreciate all who can join us today. And today we have two speakers from Cargill, Brian Lefebvre and Jason O'Brien. And I'll do a short introduction and then we'll hand it off to them. Brian has been doing biotechnology research for quite a while, and from what I understand, he loves doing it. He is director of R&D fermentation at Cargill, and he is part of a team that works to improve commercial fermentations and launch new fermentation processes. In his previous roles, he was in industrial biotechnology, also served as faculty in chemical engineering, also a member and co-founder of a startup in biophysical analysis equipment area. He has bachelor's degree in chemical engineering from the University of Minnesota, Twin Cities, PhD in chemical engineering from the University of Delaware, and his postdoc research was in biochemistry and biophysics at the University of Pennsylvania. Jason O'Brien, our next speaker, is a biotechnology leader specializing in fermentation and biochemical manufacturing. As Senior Director of Fermentation R&D at Cargill, he drives R&D and process development, enhancing efficiency and reducing costs. He's held previous roles in operations, process engineering, in a number of biotechnology processes, from large-scale established businesses to new to the world scale-up. Jason has degrees in chemical engineering and biochemistry from the University of Minnesota, Duluth. And with this, I'm handing it off to Brian. Brian, welcome, and I believe you are the first to start.
Thanks so much. Thanks for the welcome. Thanks for the invite. Thanks for the introduction. Thanks everybody on the call for attending. I did a check beforehand. I believe folks can see the slides. First slide is just our title slide explaining that Jason and I are going to spend time today speaking with you about fermentation at Cargill. There's kind of two pieces of that. It's only a 3-word title, but there's two pieces, the fermentation piece and the Cargill piece. So we'll start off with some information about Jason and myself that kind of supplements the introduction that Irina gave. And then we'll talk about Cargill, big picture Cargill, all the way down to what we do in fermentation at Cargill. So I'll advance the slide. That's me. You can see me on the video. You can see the picture of me up there. When Irina gives the introduction and you hear about yourself, sometimes you think, wow, it was a long time ago I did those things. So I started off in biotechnology. I got my bachelor's degree in chemical engineering from the University of Minnesota, and I started doing research in the biotechnology area after my sophomore year as an undergrad. I was fortunate enough to find my way into a laboratory where they did organism construction and fermentation R&D, and I just really liked it. So that kind of set my mind on the path to go to graduate school, where I went up to the Mid-Atlantic region to get a PhD, still working in chemical engineering, but on the biotechnology side of that. And afterwards went and did a little bit more in the biology side, structural biology by proteins by NMR. And part of the work I did there led to the launch of this startup company, which is still functioning, selling equipment, specialized equipment for doing protein NMR under really high pressure. So I work with some really smart people who are really good at the structural biology piece, and I kind of brought the engineering side. How are we going to design devices that allow people to do this kind of work? Before going into industry, I was a faculty member at a school in New Jersey called Rowan University in the Philadelphia metro area as well. So I spent 5 years as a faculty member there in chemical engineering, teaching and doing research in biotech and pharmaceutical area. And then was lured away from academia to go work at the experimental station in Wilmington, Delaware for DuPont, where I spent about 10 years there. First 2/3 of it was doing cellulosic ethanol fermentation research, which is a process that during my time there, we took from laboratory to pilot to demonstration scale with a plant in Tennessee that I did a lot of experimental work to inform the design and help start up that facility. And then kind of the same process over again to get plant built just outside of Ames, Iowa, in Nevada, Iowa, a commercial scale plant. So it helped a lot with the design, experimentation, startup of that facility as well. And then eventually at DuPont, also got into other types of fermentations and team leadership and competency development. And then not quite 10 years ago, came back to the Midwest to work at Cargill, still working in the fermentation space. Working here out of what we call the Minneapolis R&D Center, which is in Plymouth, Minnesota, which is a western suburb of the Twin Cities. So we have a fantastic team here, a fantastic facility here for doing fermentation research, and we'll tell you more about that after Jason introduces himself.
Thanks, Brian. So similar to Brian, you can see me. You can see my picture up on the slide deck. A little different. You see me in an office now. That was interesting day in the plant. As you can see some stuff on the wall behind me. Tying back to Iowa. So my first experience